We are going to discuss about polysaccharides, specifically starch. Now, starch is one of the polysaccharides that forms structural compounds in the body of living organisms, especially plants. Now, it has a general formula of C6H12, H10, and 0.5 in bracket n. N is a large number. Can be 5, 6, 10, depending on the type of the polysaccharide. So therefore, this is the general formula of starch. Now, examples of polysaccharides, apart from starch we have mentioned, is cellulose, we have glycogen, we have chitin, we have lignin, we have aminopectin, we have pectin, and etc. So, because I've said specifically we shall deal with starch, remember, starch is one of the structural compounds in plants in which it is found as a stored food. It's a stored food in plants, and therefore it is found in seeds, it is found in the roots in form of starch molecules. Now, cellulose is a component of cell wall in plants. If you look at plant cells, inside we have the cell membrane, and outside we have the cell wall. So therefore, it's a component of the cell wall in plants. Glycogen is a stored carbohydrate in animal tissues, especially it is found in the liver, it is found in the skeletal muscles. So therefore, it is found as stored carbohydrate in animal tissues. Chitin is a structural component that is found in the exoskeleton of organisms that are found in phylum Arthropoda, and also the cell wall of the fungal species, especially the iva. Remember, the iva is the basic unit of kingdom fungi, so therefore its cell wall is encased by chitin, and chitin is an example of a polysaccharide. So therefore these compounds, chitin, glycogen, cellulose, starch, all of them are polysaccharides. And since they are polysaccharides, you find that each unit or each comprising unit is made up of several monosaccharides. Remember, monosaccharides that are found in nature, we have fructose, we have glucose, we have lactose, galactose. All those are examples of monosaccharides, but it depends on which type of polysaccharides they form. Because during hydrolysis, when polysaccharides are broken down, they are broken down into respective disaccharides, and then from the disaccharides, they are again hydrolyzed into their respective monosaccharides. Now, what are the properties of starch? Starch has a high molecular weight. Remember from the formula C6H10 if you multiply by several number like 5 or 6 or 7, you find that you get a large molecule. So therefore it has a high molecular weight. Also, it doesn't have a sweet taste. If you taste starch, like for example wheat flour, there is no sugary taste. So therefore it doesn't have a sweet taste. So therefore it is not sweet in nature when it is tested. Now, they are also insoluble. They are insoluble in water. If you mix starch until you stir continuously, you realize that the particles will not dissolve completely in water, so therefore it is insoluble in water. Also, I say it is hydrolyzed. It is hydrolyzed into the respective disaccharides and monosaccharides. For example, during germination, when starch is broken down in the seed, this seed is hydrolyzed into several disaccharides. For example, starch is broken down into maltose. From maltose again, it is broken down into respective monosaccharides such as glucose. So therefore, it is broken down into their respective disaccharides and monosaccharides. Now, what are the importance of starch? Starch is source of energy. One is that it is source of energy. So therefore, it is oxidized to produce energy in plant tissues or animal tissues. Two is that they have structural compounds. For example, if you look at chitin, it forms part of the exoskeleton in insects or arthropods. If you look at cellulose, it is found in plant cells. It forms a protective layer in plant cells. So therefore, it's a structural compound. 3 is that it combines with proteins to form nucleic acids. Examples of nucleic acids which are formed are the DNA and RNA, which forms part of the nucleolus and nucleus respectively. Again, we have storage compounds. You find that these disaccharides, they form storage compounds. For example, starch is found in plants and glycogen is found in animal tissues. Remember I said starch, it is found either in the seeds or in the roots of plants, while glycogen is found in the skeletal muscles and also in the liver as a storage tissue. Now, if you look at starch, I said several glucose molecules combine during condensation to form that long chain. The long chain of starch is known as glycosidic chain. It is known as glycosidic chain, and the bond that ensures that glucose molecules are held together in starch is referred to as a glycosidic bond. It is known as a glycosidic bond. I know most students, they confuse between the hydrogen bond and the glycosidic bond. So in starch molecules, the bond that ensures that glucose molecules join together to form the longest chain, which is glycosidic chain of starch, is referred to as glycosidic bond. Now, how to test the presence of starch? Now, in starch, if you are given a measuring cylinder, how to describe, you say measure exactly 1 centimeter cube if you are provided with a measuring cylinder. So you say measure exactly 1 centimeter cube of the test sample. Then add 3 drops of iodine solution. Now students are fond of saying measure 1 centimeter cube of the test sample or food sample and then add 3 to 4 or 2 to 3 drops. You cannot commensurate 2 to 3 drops. 2 to 3 drops cannot be defined. So you say exactly, is it 3 drops or is it 2 drops? So you say add 3 drops if it is 3. If it is 4, you say add 4 drops of iodine solution. There is nothing like 2 to 3 drops or 3 to 4 drops. There is nothing like that. And 1 centimeter cube of the food sample is the recommended sample that will give you a qualitative test for that food you are testing. So it's a qualitative measure that gives you exact result of the sample you are testing in that laboratory. So therefore, after measuring above, because when I say above, it means you have not been provided with a measuring cylinder to measure that food sample. So I would say measure about 1 centimeter cube of the test sample, then add 3 drops of iodine solution or add 4 drops of iodine solution. Now, observation. After adding 3 drops of iodine solution, you realize that the color of iodine, which is brown, the brown color of iodine solution will change to blue-black. Don't just say the color changes to blue-black. So which color changes to blue-black? It's supposed to be the brown color of iodine solution that changes to blue-black. And when it changes to blue-black, or when that brown color of iodine solution changes to blue-black, you will say there is presence of starch. There is presence of starch. And the chemical test you are performing when testing starch is referred to as the iodine test. So I'm going to demonstrate briefly about the samples I have in front of you. Now first, this is the food sample. When I say, first of all, before you test, you start. You start thoroughly to ensure that all the particles are uniform because they are insoluble in water. Remember we said these starch molecules are insoluble in water. So there we have to start. And then measure about 1 centimeter cubed. When I say about, it's not exactly. Remember, it should be qualitative test. So when it is qualitative test, you measure about to give you the exact results. So this is 1 centimeter cubed of the food sample. Now, then I say add 3 drops. If I had 3 drops, I mean 3 drops. There is nothing like 2 to 3 drops. There is nothing like 3 to 4 drops. If it is 3, it is 3. If I measure 4, I will say add 4 drops. So here I'm going to put 3 drops. Is 1, 2, 3. Now, in that case, you shake. When you shake, you realize that the brown color of this iodine solution has changed to blue-black. This is the color of blue-black. So you will say the brown color of iodine solution changes to blue-black. Don't just say the color changes to blue-black. And then in some other cases where starch is present in small quantities in some solutions, remember, you are not recommended to shake. If you shake, that brown color, that blue-black color will disappear in most of the food samples. So examiners are very consistent in this. So therefore, they put very little starch and they want you to be very exact when it comes to observation. So you will just see a blue-black ring when the amount of starch is minimal in a food sample, especially when it is mixed with other food substances that you are required to test. So otherwise, this is the color of blue-black. The brown color of iodine solution changes to blue-black. That is all. It's the end of our lesson.